As it transitions into its second phase, the Hearing Restoration Project is building on the information and data gathered since its launch to accelerate the search for a cure for hearing loss and tinnitus. Hearing Health Foundation (HHF) recently announced the third consecutive year of funding new research projects through the Hearing Restoration Project (HRP) consortium. This year is exciting as the HRP scientists are finishing up the first of 3 phases of the strategic research plan. This plan defines the roadmap to clinical trials for a cure for hearing loss and tinnitus within a decade. Many types of hearing loss result from damage to the delicate hair cells of the inner ear. Humans cannot regenerate these cells, but in a game-changing breakthrough in 1987, HHF-funded scientists discovered that birds can. Over the past several years, HRP scientists have produced new genomic data sets from fish and birds, which show regeneration, and from mice, which do not. These datasets now allow us to take the next steps in understanding which genes promote regeneration in some animals and which genes block it in others. The 2014 funded projects will continue to move us closer to our goal of inducing hair cell regeneration in people to produce a cure for hearing loss and tinnitus, says Peter G. Gillespie, Ph.D., the director of the HRP consortium. I am incredibly pleased with the outcome of the work the HRP consortium members have been conducting over the past several years. As a person living with hearing loss, I am thrilled with the progress that the HRP consortium is making, says Shari Eberts, the chairman of HHF's board of directors. We are funding the best hearing scientists, conducting groundbreaking research, and are on track to see a cure for hearing loss and tinnitus. 3 projects have been renewed, see opposite page, and 4 are being initiated. Each brings us closer to a cure. Here are details about the 4 new projects. Bioinformatics and gene expression analysis in zebrafish, chickens, and mice as a tool to identify genes for future functional studies, led by Andy Groves, Ph.D., Tatjana Piotrowski, Ph.D., and Jennifer Stone, Ph.D. This project is focusing on the bioinformatic analysis of genetic data collected throughout phase I of the HRP. Over the past 2 years, the HRP has gathered hundreds of thousands of pieces of genomic data from zebrafish, chickens, and mice. Bioinformatics is a set of sophisticated computational tools that will allow us to compare this genomic data. Since we know that zebrafish and chickens spontaneously regenerate their inner ear hair cells, we can compare their genetic data to that of mice, which, like all mammals, do not regenerate hair cells. Once we know which genes, or series of genes, known as pathways, trigger regeneration in zebrafish and chickens and which inhibit it in mice, we will have better targets for drug therapies that may be able to induce regeneration in humans. Single-cell transcriptional profiling of chicken utricle and basilar sensory epithelium cells after aminoglycoside-induced hair cell loss. Another new project expanding on work started in phase I is analyzing the inner ears of chickens. Stefan Heller, Ph.D., Michael Lovett, Ph.D., Jennifer Stone, Ph.D., and Mark Warkol, Ph.D., are using newly developed techniques to study how supporting cells react when neighboring hair cells die and which signaling pathways are activated or deactivated. They are also determining if this new technique, known as single-cell resolution, can be used more broadly in analyzing regenerative capabilities. Characterization of DTR mouse model for hair cell regeneration research. Edwin Rubell, Ph.D., who is known as the co-founder of hair cell regeneration in chickens, is working on the creation of a mouse system in which inner ear hair cells can be reproducibly removed from the inner ear without doing damage to other components of the inner ear. Rubell's lab has designed and produced a new mouse line in which a toxin can be introduced at any age and it will reproducibly cause the rapid and complete elimination of all hair cells in the cochlea and most hair cells in the balance organs of the inner ear. This model system will allow for the systematic study of hair cell regeneration in live animals, and at any age. Expression of WNT pathway molecules and potential to stimulate hair cell regeneration. Alan Dabdub, Ph.D., and Albert Edge, Ph.D., are collaborating on a project studying the signaling molecules in the WNT pathway to better understand its role in regeneration. WNT signaling has been shown to play a major role in stem cell biology, cell proliferation, and cell fate determination. These three previously funded projects will continue through 2014 to finish data collection efforts. What stops the inner ear from regenerating? The mammalian cochlea seems to start out with an intrinsic capacity to repair itself, but this disappears as the cochlea matures. Andy Groves, Ph.D., and Neil Sedgwill, Ph.D., discovered that by blocking a specific cell communication method, the supporting cells next to hair cells allow for hair cell regeneration in very young mice. See Fall 2012's What Stops the Inner Ear from Regenerating at hearinghealthmag.com. For updates about this project, see hhf.org/consortium-projects. Groves Sedgwill. Making a map. The supporting cells next to hair cells give rise to new replacement hair cells in birds and fish, but not in mammals. However, Albert Edge, Ph.D., Stefan Heller, Ph.D., and Elizabeth Eastle, Ph.D., have discovered that in mice, non-sensory supporting cells can retain some normal function for long periods of time, making them promising targets for regeneration therapies. See Fall 2013's Making a Map at hearinghealthmag.com. For updates about this project, see hhf.org/consortium-projects. Edge Heller Royster. What would it take to seal the deal? Andy Groves, Ph.D., Neil Sedgwill, Ph.D., and Jennifer Stone, Ph.D., figured out how to grow the inner ear utricle in a dish, allowing experimental access for the first time to hair cells and supporting cells in adult mammals. The utricle may be able to teach us about the signals that drive hair cell regeneration in the mammalian ear. See Winter 2014's What Would It Take to Seal the Deal? At hearinghealthmag.com. For updates about this project, see hhf.org/hrp-consortium-projects. Groves-Sedgwill Stone. The promise of a cure for hearing loss as well as tinnitus is very real. And underlying that promise is the discovery that chickens have the ability to spontaneously restore their hearing by regenerating their damaged hair cells after suffering hearing loss. While humans and mammals do not naturally regenerate hair cells, the Hearing Restoration Project is aiming to enable just that. Watch our new public service announcement, sir, that highlights these exciting developments on the road to a cure at hhf.org/curing_hearing_loss.